coming to cellular differentiation, since you already understood totipotency, it is cellular differentiation is similar, but here we will look at what actually happens inside the cell, what genes control. Okay, means it is totipotency is the phenotypic. Okay, means you saw that from the callus you can get the root or shoot. But cellular differentiation, we want to see which gene is controlling that. Okay, is it the gene? Is it the nucleus? It is the cytoplasm? Is it the organelles? What is controlling? Okay, so obviously we know it's the genetic material that control the ability of to regenerate. Okay, so differentiation is genes. Okay, are active and the gene products are used to bring some phenotype change in the cell. Okay, this line, what it means is that there are some genes. Okay, these genes, they become activated. Okay, they become activated means the transcription, translation, protein synthesis, etc. is happening. But the end product we can see that this gene is telling, okay, this gene is telling you become the shoot. Then this gene is telling you become the root. Okay, then this gene is becoming you become the flower bud. Okay, so it is the property of multicellular organisms, unicellular organisms also. Okay, so there are examples of cellular differentiation in unicellular higher organisms and animals also. So zoologists, they have studied in the aspect of the development of the zygote. Okay, so as I told you, developing a baby, okay, that is one of the greatest experiments. No, God himself, he already knew that I'm going to do totipotency and cellular differentiation when you became a baby. Okay, so cellular differentiation and totipotency is actually what you did when you were just formed as a zygote. Okay, that one cell, it has all the information whether to form an arm, a limb, a nose, a hair. Okay, then that embryo, it formed those embryoids, remember? They divided into some stages, 16 cell or 32 cell like that, and they were all callus only. No, they were awaiting. Okay, so I have already told you that. So let's look how it's happening in animals. So first, the fusion has to happen. So you have to fertilize, then cleavage. Okay, then the zygote has to divide, has to cleave. Okay, I think you must have studied in zoo cleavage this way or that way. No, it has to divide to form the blastula. Okay, blastula is a group of undifferentiated cells. Okay, so from the zygote it divided into 2, then into 4. Okay, so on and so forth till you get this blastula stage. Okay, and these blastula stage behave like callus only. Okay, they are not yet differentiated. They are just awaiting. Okay, they are just awaiting for their direction. Then the gastrulation forms. Here is when specialization happen. Okay, zygote cleaves. Okay, then you get the blastula. Okay, these are undifferentiated. Then you get the gastrula. Okay, where you will get your hand, your leg, okay, your face, your hair, etc. Okay, then last is differentiation, where you get specialized cells. Okay, like super specialized. You know, after the cells got you become the lung. Then specialization, it will form the lungs. You know, the areola and all. Okay, that is the super specialization. Okay, so what does the cytoplasm play? Okay, so there are some experiments which show that cytoplasm plays a role in cell differentiation. One experiment was the egg of a snail. There was a lobe-like structure at the vegetal end. Okay, so there's a vegetal pole and an animal pole. Okay, just Google it. You know, you have already done in zoology. Okay, then if this lobe is removed, you will get a defective embryo. Okay, So again, there is another area which is known as gray crescent. Again, if you remove this gray crescent, there's something abnormal. Okay, so here the cytoplasm plays a role. Okay, then this is another example of a animal, amphibian egg. Then I don't want to keep reading. You just go through the note. Okay, so I will read this part. Okay. So these two experiments, the snail egg and that other one, they show that cytoplasm is responsible for cell differentiation. Okay. So in mammals, the embryonic cells are totipotent after the first cleavage till the development of embryo. Okay, so till that gastrulation stage, no, it is totipotent. Okay. The property of totipotency of egg cell is due to the homogeneity of the cytoplasm. It means that the cytoplasm is perfectly balanced. Okay, it's homogeneous, it's perfectly balanced. So in some experiment, when they cut this gray part, remember, there were some abnormalities in the nervous system. Okay, when they remove this part of the cytoplasm, there was something abnormal. Okay, so the cytoplasm has to be homogeneous and perfect. Okay. Fine. So the retention of totipotency by individual cell has been demonstrated in case of plants also. So any part of a plant, we can culture them and they will form a callus. That one we already know. And then from the callus, we can get a whole plant. Here, this one. Okay. So there's another work done on Drosophila, and they saw some genes called as homeotic genes. Okay. So these genes, they were identified through mutations when one part of the body is replaced by a structure that is found somewhere else. Okay. So this unusual thing occurs during development of the embryo. Okay. The mutation of the homeotic genes result, here this line, in the formation of middle legs in place of antenna of Drosophila. Okay, so if these homeotic genes get mutated, you use an X-ray, okay, and kill this gene here. So this Drosophila, okay, I drew a person. So this Drosophila, okay, this is its head. Instead of getting antenna, he's getting leg. Okay, he's getting leg on his head. Okay, so so these genes are very important to avoid mutations. Okay. Okay, so this one I've explained. Let me scroll again. So if you analyze them molecularly in the molecular analysis, you find that there's 180 base pair sequence present in other homeotic genes. This sequence is known as the homeobox. Okay, and this homeobox is also present in higher animals. Also, gene cloning and sequencing has been done from mice to man, and so they found homeobox in all. Okay. And we see that these genes, they function the same as Drosophila. Means if you mutate one, something abnormal happens. Okay, even if that cell is still in the totipotent stage, if you mutate that gene, in the homeobox, abnormality will form. Okay, is that clear? So what I'm saying here is, fine, the cytoplasm is perfect cytoplasm. Then all the genes are fine, okay, all the chromosomes are fine, but you target this homeobox and you just give one X-ray, okay, and you just destroy this homeobox, okay, even one gene out of all the, even one of a few of the base pairs, okay, the whole thing will not work. You will get an abnormal organism. Okay. So the homeobox is very important, okay, and throughout evolution, generation of generation, we have always conserved it, okay. Means in nature, when we were evolving, we mutated other places, okay, but the homeobox remained the same. So this is all about the homeobox or the homeotic genes of cellular differentiation. So this is the detail of what happens Okay. So what happens? Which gene, if you destroy which part of the gene? Okay. So this is in detail, the genetic detail. You can go through by yourself. Okay. In the exam, not really required to write all this. It's just extra information. So let's look at in plants. Okay. So cell differentiation pattern. This line we can see in detail in Arabidopsis. Okay. And Arabidopsis has been taken as a model system. Okay. All the studies that the scientists have done is with Arabidopsis. Because remember, I told you there are only 8 chromosomes, no? Right. Only 4 chromosomes or 8 chromosomes. I forgot. We did know in that one, that chapter, chromosome structure. So the scientists have taken Arabidopsis as a model system. So they're studying because it has a small genetic size, no? So it's easy to map, okay? I've already explained to you, no? So Arabidopsis is a model system. There are some factors which help in causing different cell type organs, etc. to originate at specific location, okay? So cell differentiation is noted during embryogenesis. So in maize and rice also they have studied, okay, and in soybean also they have studied, but the most detail is done in the Arabidopsis thaliana, okay. Okay, so why they chose Arabidopsis thaliana? Because of these small size of the plant, small nuclear genome, short generation time, and so on and so forth, okay. All these you go through. Okay, there are a number of genes involved during early and late stage of embryogenesis in zygote and somatic embryo. Okay, different cell division has been noted in somatic embryo. Many pattern genes have been identified through mutagenesis, which is showing seedling abnormalities. Okay, so what they did was they mutated some genes of Arabidopsis, and they observed that there was a mutant known as this one GN, and there was something abnormal with this region, okay, apical and basal. Then another mutant known as GK, the apical region was abnormal. Again another mutant which showed the central part is abnormal, okay. So so what you can conclude from here is that if you mutate these cells, okay, the differentiation, the totipotency, although it was already there inherent, but if you mutate these genes, it is showing some abnormal growth, no? There was something wrong with the central region, okay, in the case of this FX mutant, okay. So what can you conclude? Cellular differentiation depends on what? On the genes, okay, on the genes, yes, okay. Then let's go on. So again you can so there were other methods done, okay. They cloned some genes, okay, then inserted a probe, okay, and then they tagged some DNA, okay. Then they studied how this tagged DNA, how it works, okay. So this is just explaining you the cloning pattern, how it works. How did they identify which gene affected, okay. They tag, okay, they attach a probe, okay, like a marker, no, like you want to see an important you want to see your height, so you mark, no, from on the wall with a pencil, something like that, okay. So you mark them. So this is just showing you how many defects you can get, okay, in the embryo, okay, if you mutate these genes. So there are some genes known as housekeeping genes which are always maintained, okay, which are always maintained, but there were some functions that became abnormal, okay, even in housekeeping genes. Housekeeping gene is similar to that homeotic box, right, which was conserved for years, right, okay. But Even though these housekeeping genes have never failed throughout evolution, but if you alter some functions, okay, even the housekeeping gene will behave abnormally. Okay, so the first known mutant of Arabidopsis is bio-1 auxotrope mutant 122GE. Okay, so these are the net questions which can come. Okay, so this mutant, okay, this first mutant of Arabidopsis, remember we created many mutants, okay? We wanted to see, is it the genes responsible for cellular differentiation? So we wanted to see, then there is a diagram about the first mutant. Okay, what happened? Let's see what happened. So this mutant, no, this one, okay, this first mutant, there was a stoppage of growth of the embryo at the heart stage of development, but there was normal growth when biotin is added in the medium. Okay, so let's look at the pathway. So this is the pathway. Okay, this is supposed to be the normal pathway. Okay, so there was the bio-1 mutant, they were defective in the synthesis of this acid. Okay, then they added it was corrected by adding gene A. Okay, in the bacteria it was corrected. So here, if that gene was okay, that this gene, right, is supposed to produce this amino acid and this amino acid, you get it? But it was mutated. Okay, it was mutated and it could not produce this amino acid. But this bacteria, it corrected the defect by adding this gene A. All this is not necessary. It's all net related, no? But still, let's know. Okay. So it shows that the plant mutant can be recovered back to the normal by the introduction of the clone bacterial gene. Okay. So these genes, they came from bacteria. Okay. So what you did was, you mutated these genes. Okay, you mutated these genes. So instead of getting this acid and then this end product, okay, these end products, we don't get. So something abnormal happened, but you can correct it by extracting this gene from a bacteria. Okay, from a bacteria you extract and then you introduce it into the medium, it will get corrected. So what does it prove? It proves that it was gene A which allowed this synthesis to go to this step. Let me explain again. So you added this gene A from a bacteria. Okay, this is a bacterial cell. So what do you conclude? If at first we never got this product, these products, product 1, product 2, we never got these products. But as soon as we added this gene from the bacteria, we got the product immediately. So we can conclude that it is the gene, okay, the gene which brings about the cellular differentiation. Okay, so I'll stop here. So I'll end here because this is already very heavily loaded. It's a MS, not even an MSc, it's a net related like topic. You know, it's very tough. So you don't have to know all this, but you can at least understand that it was the genes and the DNA, okay, which affects cellular differentiation.